Hello and welcome everyone. In this podcast, we're going to talk about one repair mechanism, and that's all. And we're going to talk about base excision repair. You won't need to worry about the nucleotide excision repair, so let's just focus on this one repair mechanism. It's a repair mechanism used in eukaryotes and bacteria alike. And let's begin by drawing a strand of DNA here. And we'll label it 5', 3', 5', 3'. Okay, let's go ahead and place some nucleotides along here. On the top one, let's go CACG, ATTG. And then we'll write the complementary one below, GTG, CTAAC. Now, this is the way it should appear, but let's create a situation where there's a mutation right here. And instead of a thymine, we will have a uracil. And I'm going to box it off so we can keep an eye on it. So this is a mismatch, and if it's left unrepaired, it will be passed on to the next strands. So what happens is there's this enzyme called N-glycosylase. Now there are many kinds of N-glycosylases in the genomes of E. coli and eukaryotes, and each one will recognize a different mutation. For instance, this one, we're going to say, recognizes when there is a uracil in the DNA, and we know uracil is not in DNA, so it needs to be removed. There might be another, well, there is another N-glycosylase that recognizes when there's a thymine dimer, and would remove that thymine dimer, in a similar mechanism that we're going to outline here with uracil. And what this enzyme will do is it will come along here and it will remove this uracil. So let's remove it. And now what happens is, we'll draw our DNA again here, and we're going to have to keep drawing these bases here so we can follow them along here. Okay, so I boxed off where we have an empty place here. And then what happens is this other enzyme, which we don't need to worry about the name of it, but it will make a nick here. And I'm going to just signify that by erasing part of the backbone here. And so this is recognized by certain enzymes in bacteria, for instance. They'll use DNA polymerase 1. What DNA polymerase 1 will do is it will use a 5' to 3' nuclease and degrade all this, a portion of it at least. So now what DNA polymerase 1 will do is it will read this top strand as a template, and remember these are still here. It just degraded the bottom strand here because it recognized that nick, and now it uses this as a template again, and as it's laying down the new DNA, it brings in the appropriate bases, T, C, G, T, and then a G. This has now been properly repaired, and that mutation will not carry forward. Eukaryotes do something very similar, and so let's just write that down real quick too. I'm going to erase the bacteria part. So let's explain what happens in eukaryotes. Again, we have our mismatched pair that has been removed, the uracil has been removed, and the nick has been produced here. And in eukaryotes, DNA polymerase, abbreviated here, beta, it does something, it's a little bit more efficient in that it doesn't degrade all of this. It just recognizes that it's missing a base here, and it just repairs that one base. So let's just draw this back up here. It doesn't have to degrade everything, and it just places in that T and seals this up using a typical DNA ligase. And now, just as with bacteria, this base pair has been restored and the mutation is fixed. There's a lot in your book about DNA repair, but this is all I want you to know, the base excision repair pathway. You do not need to know the nucleotide excision pathway, just this one. Alright, if you have any questions, please let me know. Bye.